male and female insects, the traits that they use to interact, with an emphasis in his studies on the color traits and the eyes that see them, which is especially what we're going to learn about tonight. He's been doing this for 15 years. He started in Arizona, well, actually started in Cornell, right? He started doing this stuff in Cornell.
Yep.
Undergraduate research project, started studying insect stuff. Moved to Arizona and then did a cool postdoc in France. And then another thing that I thought was cool, he got a project, parts of this project crowdfunded. And that was showcased in Science magazine. It was pretty neat. So, Trisha and Ed, new faculty in Lux, are hosting tonight's social. The address is 1784 Gateway Drive, which is also 1784 North, 1700 East. So head towards Green Canyon. There's a little map over there. So, and sign-in sheets should be going around. With that, Mabel
Thanks, Keelan. And thanks to all of you for coming. I'm going to be telling you tonight about work that we've been doing on jumping spiders only for the past 3 years. So this is a really new emphasis in my research group. Tomorrow at 4 p.m. I'll talk about work that we've been doing for a little bit longer on butterflies. And so I feel a little bit schizophrenic in my lab because we have these two very different systems going. But I'm really excited to tell you about this work today. And like Keelan said, it was crowd-funded parts of it, and it was just funded by the National Science Foundation too. So yay. So, but I want to explain really quickly why I say fly tiger in my talk, because I think this is a little interesting detail. So these jumping spiders don't build webs. They're what are called cursorial predators, and so they hunt their prey on the run. And actually, the Chinese called them fly tigers, and in many ways they resemble cats of prey. So they're really interesting in their ability to find prey, identify different types of prey, and plot different strategies for how they capture prey. So these are really like tigers or lions of the undergrowth. And I'm going to tell you a bit about their visual biology so that you get a sense for how they're able to accomplish this being so small. This guy right here is about the size of your pinky fingernail, so they're tiny. Okay? Before I get talking about the science, I want to talk about the people that are behind it, though. And most of what I'll tell you about today is the result of really fruitful collaborations with a couple of very talented postdocs in the lab. Dr. Lisa Taylor and Dr. Daniel Zurich, and you can tell if you know me and my lab which one of these two people has been in the lab for longer. That's Lisa. Daniel is starting to look a bit like Lisa these days, so in terms of kind of being a little bit zanier. And there's also been, thanks to the contributions of some very talented undergraduates, M. Mayer, Zarina Amin, and Kevin Byrne here. They're the original Spider team. There have been other iterations of Spider teams since then, but the work I'll tell you about today has been their contributions. So we're a fun-loving group, as you can tell. I'm an evolutionary biologist, and for me, that really means trying to understand how diversity arises and evolves over time. And there's a number of ways of answering this question. I'll talk about other ways of answering this question tomorrow when we talk about the butterfly work. But today I want to emphasize one particular way of doing this, and that's a way to approach this question that many people have taken, and that is to try to identify a group that seems to be evolving rapidly or diversifying in form fairly quickly. And so a number of groups come to mind, for example, birds of paradise, which have really had this exuberant diversification of courtship, colors and behaviors, feather form, and we can ask why this diversity arises in this group, how these types of different forms evolve. We can also ask why this doesn't happen to other groups. So these are the frogmouths, and this is another group of birds that live in the same exact areas, right? They're living in rainforests in Papua New Guinea. This is the only 3 species in this genus here. And they all look the same. So why do we find this kind of exuberant diversity in one group, and in the same environment we're finding that other groups don't diversify in this way? And so by thinking about this in a kind of evolutionary lens or a phylogenetic lens, we're really oftentimes looking out for these groups that have undergone massive radiation of form and comparing those to other groups that have remained fairly over time or slowly diversified, or maybe even lost diversity over time, like horseshoe crabs, for example, which we only have a few species left. And we can think about the generation of diversity in two major ways that I'll get to later in the talk. The first is to try to understand how this diversification occurs via major transitions. So perhaps there's something right at the base of this group here that happened that resulted in the ability for evolution to really take off and diversify the forms that we see in that group. The other way that we can think about this is considering processes that are occurring within this group, within these clades here, to try to understand what perpetuates this diversity or this diversification process. So we're going to be thinking about this, but we're going to be thinking about this particularly with regard to visual traits. And with visual traits, we oftentimes think of a coevolutionary process between the systems, the visual systems or eyes that see these traits, and the signals themselves. So the question that we're going to approach tonight is how visual systems influence the evolution of visual signals. And I want to build a little bit of a framework for you to be thinking within about how we think this process occurs. So when we think about the coevolution of visual systems and visual signals, most people often think about a unidirectional process where something about the eyes, the way that they function or how they're built, influences the kinds of signals that work well to stimulate them. So it may have to do with their colors or contrast within the signals or movement within signals. And so we oftentimes think that eyes will shape the types of signals that work well. Of course, there's a reverse process here, so visual signals can select for changes in the way that eyes function, and this process, therefore, can be some kind of bidirectional coevolutionary process. This arrow here is much less well understood. But we need to think about this in the context of the ecology of these animals. And I realize I'm at an ecology center and I'm an evolutionary biologist. So I want to appeal to the ecology here and say that we need to think about this process as occurring within habitats, within microhabitats or within niches, because aspects of the environment are really going to influence the dynamics between eyes and the signals that stimulate them. So things like the background against which animals signal, the color, the complexity, the movement in those backgrounds will influence how well the signals are seen. They'll also influence how those eyes function, so how they pick things out of the background to see that are of interest. Things like the light that's available in the environment are also going to influence how eyes work and process visual scenes. So these components here are really essential to thinking about how the diversity of colors and pattern and form evolve in these types of groups. It's typically thought of in something called a sensory drive framework. Now, if we want to understand the influence of these different pieces of this, we'd really like for variation in each of these. So we'd like variation in the visual systems, we'd like variation in visual signals, and if possible, we'd like this variation to occur in different habitats. Right? And so one of the main reasons why we work on insects and spiders is for the very reason that they provide us with the requisite variation that we need to begin understanding how these things are linked. I'm going to be telling you today about jumping spiders here, the colorful ones in particular, but these apply to insects as well. And I want to talk to you first about variation in visual systems. In these groups here. So there's an extraordinary diversity in the way that eyes are built. And I just want to highlight a few things that you may or may not know about here. This is a whirligig beetle. How many people know what a whirligig beetle is? So these are beetles that live on water surfaces, and they've actually split their compound eyes into two eyes, one that functions above the water and is actually built to see well through air. So the optics of the lenses of this eye are built to look through air, whereas there's one that's always submerged under the water, and it is built to see well through water. Right? You can't do both things well at the same time. That's why you have a hard time focusing underwater. And so they've split these functions here. Here's a mayfly male right here, and they have likewise split their eyes up into functions that allow them to do different things. So they have these side-looking portions of their eyes here that help them to see the horizon and features of the horizon in the world around them. But these turbinoid eyes right here that point upwards are incredibly sensitive to movement as well as to form, and their function is to see females against the night sky or against the twilight sky. So they've split their functions up in their eyes as well. to try to maximize their ability to see things. Spiders are also incredibly fascinating, and after you leave tonight, you'll know way more about the way spider eyes work than you ever thought you might. Spiders have 8 eyes, typically, and they are single lens eyes that have evolved to have all sorts of specialized functions. So this is a wolf spider right here. These eyes here function to see long distance, and they identify prey at many body lengths away. These eyes here actually have their focal points focused right between the jaws, and they aid in the processing of prey here. So they have far-sighted and near-sighted vision here that allows them to do 2 different things about their predatory biology. Now, in contrast, the vertebrate eye is fairly conserved across a wide variety of groups, from birds to mammals to fish. This is a single lens eye, and of course there's variation in these eyes, but not nearly the kind of variation that you see here, which makes these groups much more compelling to study. Okay? And so we have diversity in color and form, but also in eyes. The other thing I want to point out, and this is kind of a neat graphic that came out of the National Geographic magazine recently, is that although we know there's immense diversity in these groups of animals here already, we're just at the tip of the iceberg. So in contrast to mammals, where we think we've discovered roughly 98% of the species that exist, for insects we think we only know about 20%, and for spiders we only know about 17%. So not only is there a lot of diversity out there, but there's even more than we know about already. So these make these groups really fertile territory for studying this kind of thing. So I'm going to tell you today about our work to try to understand how a group of really brilliantly colored, 2 groups actually of brilliantly colored spiders go about seeing, using color, and some of our preliminary results about how their eyes and their displays may be co-evolving with each other. And we do this work in 2 major groups here. There are 2 genuses or genera. One that's North American. These are called Habronatus jumping spiders here. And I failed to look up whether or not you have them in Utah, but I'm quite certain you have at least a few species in Utah. We mostly work in the arid Southwest, which is a biodiversity hotspot for this group here. And we're also beginning to work in collaboration with Damien Elias and Maddie Gerard on the peacock jumping spiders that some of you may have seen some of the YouTube videos that have started to emerge. These spiders, you're looking at only males right here. Females are fairly drab. And the males do these wild displays to try to impress females. So this is one of these peacock jumping spiders here. And not only is he doing all sorts of movement and displaying a very colorful abdomen here, he's actually also singing to the female. And in both of these groups, males sing. They have stridulatory organs between the head and the abdomen here that function a lot like cricket song, but transmit vibrations through the substrate to the female who hears them with her legs. Right? Now you can see the male's very gentle and cautious here. The reason being that the female is pretty likely to eat him instead of mating with him. If you want to impress friends at a cocktail party, you need to remember this. It's called pre-copulatory sexual cannibalism. And this is a real issue for these males. These females are voracious. This male got lucky here. But he has to go over her jaws and turn her abdomen to get access to her genital openings called gonopores. And so males are incredibly cautious. They're really dancing for their lives in these species here. Okay? Now, why are we just beginning to learn about the biology of these animals here? And the answer is that it's been very difficult to get at the biology because they're tiny. They're incredibly small. This is a penny. Okay? And here's a spiderling right here. The male you just saw dancing is about 3 millimeters in total body size. So these things are incredibly small. They're in many common habitats. So we have them in Pennsylvania. We have them close to the University of Pittsburgh. People are totally unaware that they're there because they're so tiny. And the video that I just showed you has only really been possible in the past 5 years with major advances in macrophotography and videography that allow us to to film these animals at this kind of scale. Measuring their colors and measuring their eyes has also been inaccessible until very, very recently. So fortunately for us, the technology and the interest in these animals have kind of coincided, and we're now at a point where we can begin asking interesting questions about this really fascinating group. So tonight I want to talk to you about four main things. Here's my outline here. The first thing I want to talk to you about is how jumping spiders see. Then I want to tell you a bit about our work on how jumping spiders use color in their daily lives. I want to talk to you about how jumping spiders see color. And then we'll get into some of the coevolution that I was talking about earlier. My introduction was a little bit aspirational in that we're just beginning to work on this stuff. And so I'll give you some sense for the directions that we're taking with this. But we don't have a lot of solid results here yet. Okay, so let's start with how jumping spiders see. Jumping spiders are actually incredibly visual animals. One of the first things that you notice about them is that they're working with the world around them by looking at things. And so if you've ever had one in your kitchen or on your windowsill, you'll notice that they almost immediately will swivel to look at you once they've seen you. And because we're very visual animals, it lends them a very clear sense of intelligence. They're very visually intelligent animals. And one of the reasons for that is that they have 8 eyes that provide them with extraordinary visual capabilities. Most of the inside of their head is retina, as it turns out. So that in terms of nervous system tissue, somewhere between 50% and 60% of their investment in neural tissue goes into their retinas. And they have a poppy seed-sized brain that has to do all the integration, and we know nothing about how that works yet. So they have 360-degree vision around their periphery, and that's provided by these lateral eyes that I'll be calling secondary eyes today. Those are highly motion sensitive and colorblind, and I'll tell you a little bit more about them. They have a couple of really extraordinary eyes right in the front of their faces here called the principal eyes or anterior medial eyes that have very limited fields of view, and I'll describe why that is. And then they have a couple of upward-facing eyes that are here. And those are they're not really image-forming. They're there to have them respond to predators that come in from above. So it gives them a little bit of an advanced warning system. But when they see things in the back here, they really do have eyes behind in the backs of their heads. They swivel very quickly to try to train this frontal field of view on the object of interest, because this is really where their vision is most capable. I want to talk about these eyes on the side here, the secondary eyes first. When we cut those in cross-section, we find that although they have small lenses, their retinas are right up against the lens here. And that gives them a fairly large field of view. So the combined field of view of those two side eyes is very broad out in front of the face. That's this gray wedge here. And they actually have really quite extraordinary vision in those for a small animal. So remember that these animals that I'm talking about right now are the size of your pinky fingernail. And in the center of those eyes, they have an acuity of 0.4 degrees. And I'll give you a couple of rough landmarks to bookend that. This is between a dragonfly. Dragonflies are incredibly competent predators, very visually guided, and a rat. This is actually about as good as a rat gets, okay? And so these animals have pretty good vision in these side eyes here. So they have medium resolution in this broad field of view out in front, but they're colorblind in these eyes. They are only green sensitive. They have large photoreceptors. Each one of these little dots is a photoreceptor in the retina. And those are large and very sensitive to light, and so that allows them to have motion sensitivity, really quite exceptional motion sensitivity. What about these eyes here? You can see that the red here is their field of view of these large eyes right here, and I'll talk about these little arrows in just a second. These eyes are, although they look like they should have a bigger field of view, they actually have a much smaller field of view, and the reason for that is that although their lenses are really large, the retina is this tiny thing all the way back here, and it's positioned at the end of a very long, what's called an eye tube here. And the reason for that eye tube is an advanced set of optics. So they have this large corneal lens out here. This is a converging lens, but at the very back of this clear fluid-filled area here is a diverging lens called a pit lens that magnifies light or the object onto a retina down below. And so how this works is you have a twin lens system here that actually magnifies an object in the world around on the retina here. And this is how telephoto lenses or Galilean telescopes work. So they really essentially have a Galilean telescope that's attached to the front of their head. Okay? The retina itself is also unusual. Light comes in through this pit lens up on the top. These retinas are boomerang shaped for reasons that we don't fully understand at the moment here. But they have 4 layers of light-sensitive cells within their eye here. The two top layers here are kind of sparsely populated and mostly have cells right in the center of the fovea of these retinas, whereas the bottom 2 layers here, tiers 1 and 2, have these photosensitive cells across the entire boomerang, and they're incredibly densely arrayed here. Okay? So let's give you some sense for how this works. You've got this paired lens system that works like a telescope, magnifies things in the world around. You have this incredibly densely dense photoreceptor matrix in the eye, and the result is extraordinary vision. And I want to place this in the context of other animals and how their eyes see. So this is a classic relationship between body size, this is represented here by body height, and the ability to resolve differences or pattern in the world around. So lower values of this here mean better spatial acuity. And it's really this classic relationship, which is that in order to have a good eye, you have to build a big eye. That's why big digital cameras get better images than your cell phone does, okay? And it has to do with physical limits here. So here's a beetle right here. There's a fruit fly, and they have really poor vision. Here you are. You've got great vision. You've got pretty exceptional vision. And one of the things you'll notice is that these things follow along this line here, with some exceptions. So here's a falcon right here, and of course their predatory lifestyle means that they need to be able to spot very small things at great distances. So little mice running around in the field, they want to see from high up in the air. And so they come off this curve here because they've really invested in exceptional eyes. So where do our animals fit? Well, here is the retina that I already showed you. Here is that principal eye retina here, and we get extraordinary visual acuity here. So there's the secondary eyes. So they're off the curve a little bit here. Here are the principal eyes. So these animals, even though they're tiny, they actually have the ability to resolve pattern in the world around them better than your cat. Okay? They can see the world better than your cat, and they are smaller than your cat's eyeball. Their entire body is 1/10 the size of your cat's eye. They're about as good as a pigeon or a chicken, okay? But they're incredibly small. So this is an amazing feat here. And that's enabled by this telescope-like function of their eyes. The drawback to this telescope-like function of these eyes is that they have an incredibly limited field of view, like a telescope. They can only see a few degrees of the world around, okay? So how do they solve this problem? They can only see a little bit of the world around them with these eyes. Well, they solve it by using muscles attached to this tube here to move the retina behind this big lens to look at the world around them. And I want to show you what this looks like. It really feels like you're seeing these animals think. So here's a spiderling, and you can see that it's moving these eye tubes around, trying to look at this brush right here. Okay? As spiderlings, these things are oftentimes transparent. Just to give you scale, there's a fruit fly. Okay? So these things are incredibly tiny. And so within the head, they're moving those retinas around. And everybody always wonders what this little red dot is in here. It's something it's eaten. And it really is the case that everything's connected inside here. So as it moves these retinas around, its gut is kind of squishing around. Things in the gut are souping around in there. Okay? Now, when these animals grow up to be large, they're no longer transparent, so we can't look at their retinas inside their heads while they're looking at stuff when they're adults. And we're interested in the adults. We're interested in where these animals look as adults. And so what we do is we use an ophthalmoscope, similar in principle to the one that you might have seen at an eye doctor, that looks in through the lens into the eye and images where the retina is with infrared. And we can do this while we're playing them videos. Okay, so what you're going to see next is those boomerang-shaped retinas following around this video. So they're watching they're following this little dot on a screen here, and we can image where their retinas are moving around to see how they respond to things. Live animal looking in through its eyes. So this will become important a little bit later. Okay? The other thing that I want to bring up about these extraordinary eyes is the tearing of their retinas. And so people have puzzled about why they have these retinal tears. And it turns out that these lens optics here have a very strong chromatic aberration. And if you haven't taken physics in a while, this just simply means that they focus different colors of light at different depths behind the lenses. So they focus short wavelength light, UV and blue, closer to the lenses than they do long wavelength light like green and red. And so the thinking behind why this happens is that they are focusing the light these layers are sensitive to at the appropriate depth within the eye. So what they do is they put their UV and blue sensitive cells up closer and their green and red sensitive cells a little bit farther away. And this solves the chromatic aberration by just using a tiered retina to do this. Well, it turns out that's partially true. Okay, so when we look at this is immunohistochemistry here for the UV photoreceptors, and in these upper two layers of the retina, that's where they're UV sensitive. Okay, and the bottom two tiers, they're green sensitive. So they roughly do this kind of adjustment. So they put their UV ones up here and their green ones back here. Okay. So to integrate this with our work, we know that their frontal field of view has this color-blind but very motion-sensitive vision out here with an area within which these movable retinas can travel to fill in color and detail. Okay? So how do jumping spiders use color in the world around them? We're going to be talking about this in these animals right here. This is the fiery-haired jumping spider. We're starting to invent actual common names for these things because they didn't exist before, but Pyrethryx means fiery-haired. This is a male right here, and you can see that the male display has lots of color in it. Orange knees, bright red face, and green legs. Here's what females look like. So they're pretty drab, boring looking for most of these jumping spiders in this group here. We collect these things in the arid Southwest. Many of them live in the Sonoran Desert, which is where I did my PhD, and it's lovely to have an excuse to go back here. And they're oftentimes in rock scree habitats like these open Ocotillo forests here, where we collect them one at a time by hand in little vials, okay? They can be in fairly high densities in these environments here. And males display these colors to females when they mate with them. So here's a male display right here. This is the male, and here's the female. Males start off with this long-range display where they're waving around trying to attract the female's attention. When they move in closer, they do this kind of stand and deliver thing where they do these weird knee pops. They're singing to these females at this point in time with these vibratory songs. And then they transition to this late stage where they do a little bit of air drumming here with their knees held up. So these are actually really amusing. Each of these species does this slightly differently here. And so this is an obvious place for them to be using color. And it turns out that they do care about color. Females do care about color. So my prior postdoc did an experiment where she looked at the role that this red face plays and how close a male can get to a female during courtship. This is a surrogate for how well he's doing because she'll chase him off if she's no longer interested. And what she did was that she took males that had the red face intact, and then she actually removed the red face on some males, and she did this with Sephora eyeliner, liquid eyeliner. I kid you not. She worked with a makeup artist to try to figure this out because these animals are incredibly sensitive to environmental toxins. And eyeliner, actually, it turns out, is in very low toxic, very non-irritating vehicles because you don't obviously want to put something irritating on your eye. And so she blacked out the face on some of them. And what she found was that males that had a red face were able to get closer. They were able to get farther down the road of their courtship to females than males that had that red removed from them. But this only happens in full sun. And this will make sense a little bit while later. So this will become a theme. Just file that away. So in full sun, females seem to care about whether or not males have a red face. Okay? So it appears that at least the red face we know is playing a role in courtship, but of course that's not the only place that these animals use color. They're active predators. They eat a lot of prey items each day. We feed them several crickets a day, and they probably eat somewhere close to that each day in the field. And so we wanted to know whether or not they were making decisions about what to eat based on the color of the prey that they encounter in their habitats. There's lots of small prey items that are incredibly colorful. So this right here is a harlequin leafhopper. And if you've ever encountered one of these things are 2 millimeters roughly, 2 to 3 millimeters. And it's not plausible, at least to me, that birds are driving this color here. These things are barely even a snack for a bird. Many birds would not even be able to see this thing. So there's lots of color diversity in prey items that are likely to not be the subject to predator pressure from birds. Things like milkweed bugs as well. So we wanted to know whether or not some of these colors were being used in communication with these jumping spiders. And we did an experiment which we now call the cricket rainbow experiment, where we asked jumping spiders to taste the rainbow. And what we did is we took what are called pinhead crickets, first nymph instar of a cricket here. And it turns out they're translucent enough that if you feed them colored water, they take it up in their gut and it changes their personal appearance. Right? So these are like designer crickets right here. And we put jumping spiders in arenas with these colored crickets, and we looked at the rates at which they attacked different colored prey. We did this with wild-caught jumping spiders that are coming in from the field. And it turns out that they attack red and yellow prey items at lower rates than they do blue items and maybe green or undyed items. So there's a clear bias in how they're attacking prey. This is only in full sun. So again, the theme here is that color seems to matter in high light conditions. But nevertheless, these animals seem to be making some decisions about what to attack or what not to attack based on color. First evidence, to our surprise, first clear evidence of the use of color in prey decisions for any invertebrate predator on land. So we've never seen this before, which was a shock to me. Okay? Then we said, well, can they learn color? Can they use color as something that they use to discriminate prey and learn that some prey are no good and some prey are delicious, right? So what we did is we taught spiders to either learn to avoid or to prefer red prey. And we did so using these little milkweed bugs here. It turns out that milkweed bugs are incredibly toxic if they're given access to the toxins in milkweed. So you can feed them, you can raise them on milkweed seeds, and they sequester the chemicals in the milkweed seeds. But they're actually perfectly happy to come through on sunflower seeds. And that means they have no toxins whatsoever. And so these ones are, although they're bright red still, they're perfectly palatable. And so we taught a population of jumping spiders that the red was either toxic or was perfectly fine. And then a third control group had access just to fruit flies, and white-eyed fruit flies, mind you, because we didn't want them to learn that the red eyes that fruit flies typically have were fine. And so we didn't want them to do any color learning in this environment here. Okay? And so then what we did is we offered them a choice afterwards of either a brown cricket or a red cricket. We wanted to know, have your experiences with other red prey items influenced what you're willing to attack? So here's the proportion of attacks, and here is the red aversion treatment, the red preference treatment, and the control treatment. And what we find is that in the red aversion treatment, they attack red crickets at far lower rates than the brown crickets. The red preference treatment is the opposite. Right? So they attack red prey items more often than they attack brown prey items, and when they just get fruit flies, they have no preference at all. And Lisa says that this is the most beautiful data that she's ever produced as a scientist in her life. So they're using red, they're using the color red, and potentially other colors to learn what is worth attacking and what's not. So not only do they have biases coming in from the field, but they can adjust those plastically with prey. Okay, so we think that many of these colors that are in the undergrowth are actually being driven by communication with invertebrate predators like these jumping spiders. Okay, so how do jumping spiders see color? Why am I asking this question now? I told you a lot about their vision, but for those of you that were paying careful attention, I told you that they have what's called dichromatic vision. They only have UV and green sensitive cells in their retinas. This is what we knew going into this, was their vision. And what this means is that these animals are like deer or cats or dogs. We would call them colorblind. So they actually shouldn't see any difference between red or orange or yellow and green. You could hunt jumping spiders and blaze orange and they wouldn't know, right? Okay? So we decided, well, hey, there's a lot of color, and we have behavioral evidence that suggests that they can see color. So how are they doing this? And specifically, if you were to not have some way of seeing color, this is what that display would look like. So if you remove the ability to see red specifically, all of that red goes away, right? And this might be the experience of some colorblind members in the audience. So these two might not look that different to some of the colorblind males in the audience. Sorry, guys. So what we did is, after spending a number of weeks out in the field in Arizona in incredibly bright sunlit conditions, we went and sat for 2 weeks in a dim red-lit room in the outskirts of Baltimore. So this is the yin and yang of science, right? Where we were measuring the sensitivities or the absorbance properties of individual photoreceptors in these boomerang-shaped retinas here, using a technique called microspectrophotometry. Another, $50 word there for you. Okay, and so what we were trying to do is figure out, well, do they have red-sensitive cells in the eye? So we measured a bunch of things about these telescope-like eyes here. We measured the transmission of light through the lens and this vitreous humor here, which is this fluid-filled space, much like your vitreous humor in your eye. Those are both fairly transparent. And then we started measuring cells in the retina, and we get UV cells in those distal tiers here, and we get green cells in those lower proximal tiers here, tiers 1 and 2. We already knew this. So at the end of 10 days of sitting in a dark room for hour after hour, we were really frustrated because there's no evidence here that they could see red. And so we're staring at these things, these preps over and over, and then our collaborator, Tom Cronin, would just flipped on the white light. You work with these sections in dim red light because you don't want to bleach out these cells in here. You want them to remain unbleached by light. But he flipped on the lights, and what he saw was this little red spot here. And he said, there's something going on here. You guys got to take a look. And it turns out that what these spiders do is an incredibly clever thing to get their color vision. They put red sunglasses in the middle of their retinas. Okay, so this is a long-pass retinal filter inside their retinas here, and it sits right between the two green sensitive layers here. So it's positioned right here in the middle of the retina. And what this filter does is it prevents anything but red light from arriving at these sensitive cells down below. So remember, light comes in from above, it gets filtered here, and so the only thing that arrives at what are green sensitive cells down here is red light. And what this does is it creates a third color channel that's only red sensitive. They never see the green light out here because it doesn't arrive at that cell. And so they get a third color-sensitive cell type out of this, which is trichromatic vision, which is what your entire world of color comes from. Okay? You have 3 different cell types in your eye that give you your color sensitivity. The consequence of doing this with this filter, though, is that this is only sensitive enough to see color in bright light conditions. This was this aha moment. We kept getting this behavioral evidence that they didn't care about male red faces, they didn't care about the color of their prey items when it was cloudy or they were in lab conditions, only under bright sun. And the reason is because their color vision is light limited because of the way that they accomplish it. Okay? So why is this helpful to these animals? Well, we measured a bunch of the colors in the male displays here. And if you look at these colors using models that model how they see these colors with just the green and UV, they're only able to tell apart the face from the legs and the face from the pedipalps, these little cream-colored pedipalps in the front here. Okay? If you add in that third color channel, all of the differences they can see are bolded here. They're able to see a bunch of other differences within this display. So suddenly, the third legs, those orange knees, become different looking to them from their first legs, which become different from their face. The first legs are different from the pedipalps. So suddenly, they're able to discriminate a bunch of additional colors out of these male displays. So this gives them the ability to discriminate not just prey, but also to see differences in these male colorful male displays. Okay? So I want to talk a little bit about the work that we're doing to try to understand how this unusual visual system that I've just described has influenced the kinds of displays that these jumping spiders engage in. And I've really only talked to you about this fiery-haired jumping spider, Habronattus pyrethrex. But this group is actually full of differently colored animals. Okay? So these are a bunch of different animals, Habronattus americanus here, a red, white, and blue one. We had to call this guy right here, this is Habronattus halleni. We call him Mr. Sparkle Pants because he has iridescent legs that he flashes. I'll show you a video of his courtship in a little bit here. This is Habronattus virgulatus here. And the diversity is extraordinary. This guy lives in just less than a mile from my lab in Pittsburgh. I love this Habronattus pugilis, the boxing jumping spider with its little curly-Q eyebrows here. And this guy with the little blue Hitler mustache here is a new species that we've discovered at one of our sites here that doesn't even have a name yet. So we're adding new species to this group all the time. Okay, so I want to harken back to how I started this talk, which is to talk about what generates diversity here. And I want to talk about the two ways that we're aimed at this. The first is major transitions, and I want to talk about that for just a moment here. The major transition that we're looking at here, we began to wonder, well, what is the phylogenetic extent of this red filter? This is what gives them their color vision, and it's fairly easy to spot by sectioning eyes. So we've sectioned the eyes of a bunch of different related ones. All of the animals in this genus Habronattus have it, but all of the closely related animals do not. Okay, so this is a major transition in visual function that seems to be basal to this group of jumping spiders. And so what we think happened here is that the red filter arose as an evolutionary novelty. It likely provided them with fitness benefits in the context of better or more effective prey discrimination. And then it allowed for this whole world of color to evolve in their courtship. And so this really has allowed this group, which is known for its rapid radiation, rapid speciation, to diversify rapidly in terms of courtship and the visual aspects of their courtship. The other thing I want to point out is that here are the peacock jumping spiders here that I talked about much earlier. They don't have this red filter. And we're working to try to understand how they see color, but it's likely to be a totally separate mechanism. And our evidence is pointing at the moment to something called an opsin duplication event, which is how you arrived at having color vision, which is that one of the visual pigment molecule that allows you to see color, the gene for it duplicated and allowed the second copy to be selected to become different. That's where you get your red color channel from. So we think we have at least two independent transitions to color vision in this group of animals, and they have rapidly speciated in terms of bright colors. And so we're really interested in how these major transitions have these downstream effects on diversification and the color palette that these animals use in courtship. Okay, what about processes within these clades that might generate diversity? Remember that we're thinking about this in terms of how the visual system influences the signals and also about the habitat light environment and how that influences this process here. And what we're doing is we're looking at this unusual frontal field of view with these movable retinas in the front of these animals. How does this visual system interact with all of the color and movement in these displays? And we're able to get at this with that eye tracking technology that I talked about much earlier. So I just showed you a video of a little black spot being moved around in the video, and those retinas tracking that. I'm going to show you where females look when they look at a male display. Okay, so we've played this video here, and each of these is a different retina. They're independently controlled, and they're kind of looking around on this male display that they're watching. So they're kind of watching Spider TV here. Maybe Spider Cinemax. It kind of depends on how you want to think about this. They're curious about these wrist flicks up here. This is where the resting posture of these retinas will be over here when they're bored. They're no longer interested. Watch what happens when the knees pop. Oh, they pop over and they start exploring these. Now, these knee pops are totally out of the field of view of these retinas. So what they're doing is they're seeing them in those secondary eyes. Those secondary eyes are saying, hey, something's moving over here, and that cues their gaze to move over and look at those knee pops. Now, the orange colors on the knees, they can't see that at all in those secondary eyes. Remember, those are colorblind. So in order for these animals to see the orange on these knees, they have to look at them with these retinas. So these displays are collaborating movement that cues these gazes to look over there with color so that they're sure that females look at the colorful areas of their display. And so we get retinal traces like this where, this was that last set of sequences here where those knees pop and the female eyes start exploring them rapidly over here. And if you look at longer periods of time where these females are looking at male displays, you begin to get a sense for where females look when these male displays occur. And the fairly simple conclusion is that they should put color where they are trying to capture female gaze and keep female gaze. So these are hot spots for areas where they should have colorful display elements because this is where females are looking with their color vision. And so we think this is actually going to help us to understand really broad patterns about the architecture of these displays across this group. These displays seem to be to function to try to capture and retain these gaze movements and manipulate where they look within the displays so that females are able to see the specific elements of the male display that they want females to look at. So I want to play you a few videos here of some of the diversity in this here. Here's Mr. Sparkle Pants right here. He has this unusual task of needing to deal with the fact that his bright colors are iridescent. And so that means they're angle dependent. So he does a lot of these flashing displays that flash this bright color on the underside of his legs here. This is very reminiscent to me if you've ever seen these videos of the birds of paradise where males flatten their bodies out to get these iridescent displays. And so he does these weird sidling displays where he's got this big field of iridescent color. And we're really interested, again, at where females look when he's doing this. Is he capturing and retaining her attention? And does that have fitness benefits for him in terms of his ability to finish the deed, if you will? Here's Habronattus hirsutus, the hairy jumping spider right here. You can see why he's called that. He's got these tufted things, and they have these jerky displays. These animals are arboreal in their habits. And here's Habronattus virgillatus in high speed here. So this is 300 frames per second because some of these movements are so fast that we can't actually even see them at 96 frames per second, which our normal cameras allow us to do. So he does these kind of crazy jerking displays that are incredibly rapid here. Where are we going with this work here? We're beginning to think about not only where females look and how the visual signals manipulate where they look, but also how the habitat light environment influences this. And so one of the nice things about this group is that they inhabit a really wide range of different habitats, from leaf litter to rocky screes to montane forest clearings. And so we're working across this group, a large set of species, about 20 species, to look at the different displays that these animals put on. And we're interested in how their habitat shapes the kinds of things that they do. So here's some sampling. Those 4 species that I just showed you there, these are the habitats that they inhabit. Here's Habronattus hirsutus right here is arboreal. It's up in mesquite bosks, up in the mesquite trees and other kinds of bushes in those habitats. Habronattus virgulatus is out in rocky red screes here. And then a couple of leaf litter species, Habronattus geronimii and Habronattus pyrethrax here. And you can see that there are differences in the background color of these habitats. There are also differences in their movement of objects within these. And what we find is that aspects of their displays are really different. So if we look at how big their waves are, so the amplitude of their waves versus how quickly they wave their legs during these displays, which is their wave velocity, the species have adopted very different patterns of movement. And we're really interested in how this is influenced by things like the movement of the materials in the background. So rocky scree, of course, rocks don't move in the wind, but grass certainly does. And so they may be working to try to be seen above background movement noise. And so this is some of the ways in which we're thinking about the ecology of these animals. So we're taking videos at spider eye level using optics on our cameras that are really particularly tuned to their ability to see in these environments in terms of focal length, et cetera. And this should help us to understand how these animals are adapting or being selected to have particular display types that match some of the aspects of their environment. So I hope that I've convinced you today that this group of animals here, insects and jumping spiders, have a lot to tell us about some of these basic processes that may generate biodiversity, diversity in courtship, diversity in vision, and with that, I will just acknowledge a bunch of collaborators here, a great team of undergraduates, the University of Pittsburgh and NSF for funding on this, and of course the spiders themselves. And I'd be happy to take questions. Yeah.
I'll ask a naive question just to start things off for you. But so the colors drive evolution maybe a little bit, but when behavior says, what is it about color that drives the evolution of diversification of the species, even more so than just diversification of the way they move their legs and so forth, you know? And so why couldn't you get the same kind of diversification in a world without color?
That's a good question. So, so what we think is that, well, the way that I think about evolution typically is that it's a process that requires friction, right? So it requires some traction that allows for selection to drive differences between populations or species. And so the way I think about this is you're absolutely right. These animals could diversify in movement or in achromatic, non-color-based aspects of their display, and this could drive speciation for sure. But we've added in this element that they can see differences in color, and that's additional grit, if you will, in the process of speciation. So they can now see differences in color that they couldn't before. This allows for innovation and change in terms of color, that allows for differences between populations to potentially arise more rapidly. You're not asking about this, but I'm going to I'll add a little element to this, which is that we know they're using color in the context of prey decision making, and they can particularly learn to avoid red, and they seem to have an innate bias against red, and we see lots of red as part of the colors that these animals use in their courtship displays. And remember that bit I said about pre-copulatory sexual cannibalism? Males are at high risk of being eaten during the mating or courtship process, and it may be that red actually has great benefits for staving off female attack, for reducing female appetite, saying you are not in a situation where you should think about eating, you should think about me, I'm great, and you want me to mate with you, right? So there may be these cross-contextual effects, and so my prior postdoc, Lisa Taylor, is now working to try to understand those. So the experiment where we trained these animals to avoid or prefer red prey, she's now working to see if that has any influence on female preferences or female likelihood to attack males. So if we teach females that red is tasty, are they more likely to attack males with red faces than if we, and if that's true, then What that means is that the prey community that females experience influences their preferences, and that can drive rapid speciation because prey communities can be really variable from year to year or from site to site or population to population. So I know that's more than you bargained for with your question, but I think that color may actually be a very volatile, color preferences may be very volatile for those reasons, and that might be why they're distinct. Yeah.
I might have addressed it and I just missed it, but do you have some sense for answering the chicken and egg question? Like, if it seems like for prey selection, when color vision evolved, the prey must have already had color, maybe in response to a different predator in order for that to be valuable. But did color use in courtship evolve as a result of the development of color vision?
This seems to make the most sense to me logically, because it would be unlikely that the males would experience any benefit from having bright red in their display if females couldn't see it, right? And I think you're absolutely right in the way that you're thinking about this. So jumping spiders overlap with other kinds of predators that can see color, things like birds, so they do attack some of the same prey items that birds do, and so those may have evolved to have colored defenses, the prey, beforehand. And so then suddenly having color vision as a jumping spider provides you fitness benefits in prey. And it's after that event that it becomes meaningful to have color in your courtship displays. So yeah, that's the way we think about it. Of course, it's very difficult to reconstruct these scenarios, but that seems to be the most parsimonious or logically sound way to think about it. Yeah. We do see red in other jumping spiders, but it's almost always on the upper surfaces of their body, on their abdomens, on parts of their body that face away. They're not used in courtship or other kinds of communicatory interactions within the jumping spiders. It seems to be oriented to predators that come in from above, so birds and things like that. And it's really these surfaces that are displayed during signaling interactions where the color comes in these animals. So. Yeah.
So I just keep thinking about the equipment you use to see what you see. And you mentioned some of the things that you do use, but how did you get the cross-section when you showed us the 4 eyes that they have? How did you get that cross-section, and then how were you able to track female retinas when you go from males?
Okay, so the cross-sections, that is kind of a traditional histology approach there. So we're sectioning those animals either embedded or in the case of measuring their retinas with the microspectrophotometer, we do that with cryosectioning. So those are frozen sections because we need the tissue to be fresh there. And the embedding process and fixing process eliminates our ability to measure the wavelengths of light those cells are sensitive to. It also, interestingly, removes that red filter. So one of the reasons why we're probably the first to see it is that everybody else has embedded their sections, and so it disappears in the embedding process, and they were unaware of it. Your other question had to do with the eye tracking. And so the way that works is that you have 2 light paths in this ophthalmoscope. One that images the retina, and that's an infrared light path. And so it's looking at retroreflection off of the retinas in the infrared. Okay? And the other one is in the visible wavelengths to us. And that's just a computer screen that's projected through a different light path. So you project onto the retina a little video in non-infrared wavelengths, but then you have a separate source of information about where the retinas are inside the head. So we're peering in through the lenses of their eyes to see where their retinas are behind the lens. And we have to calibrate it a little bit using those little boxes and stuff to make sure that we know how all of the setup is lined up. And these are spiders. It's kind of funny. They're spiders that are suspended. So they have a little top hat, and we wax them to a little suspended thing, and then we give them a ball to hold so that they feel like they've got their feet on the ground. And so because what they want to do when they're looking at things is they want to swivel around, to see things. But what they do when they've got a little ball on their feet is they just run as if they're like on a little treadmill and feel like they're going somewhere when they're not. So probably a frustrating and disorienting experience for the jumping spiders, but we have to have them stay put to be able to do these measurements. So, yeah. Yeah.
So do some of these spiders see near infrared then? Is that, it looks like it's going up to 700 on one of your graphs.
Ish. They see farther into the red than we do. But I would say infrared is probably a stretch for what they can see. So, yeah. Infrared, so part of the cutoff there, I would flip back, but it's a ways back. Part of the problem there is that lenses oftentimes absorb infrared. Lots of tissues absorb infrared, and so it's very difficult to create imaging systems in the infrared with biological tissues. Even pit vipers that have these infrared organs, they're terrible at image formation because lots of stuff absorbs infrared, and so it reduces the image quality. So a lot of the cutoff there is probably that they're just not able to see the infrared very well. Yeah. Any other questions? Yeah.
So I don't know too much about arachnid population, but I was wondering if these complex communication and recognition system or visual systems, if they play a role with this genus in like preventing interspecific copulation.
Yeah, so that's a good question, and we haven't looked. There's a number of predictions that would come from that. So they're almost certainly using this for species recognition. But one of the predictions is that sympatric species would be more divergent from each other to aid in making sure that process happens. We haven't looked at that. There's a lot of sympatric populations, lots of populations of mixed species compositions. So that's something we'd like to get into. Males are not particularly choosy about what they court. Yeah, so, but males are incredibly sensitive to female behavioral cues. And so I think the males typically leave the discrimination up to the females, but they'd rather not the discrimination be, oh, you're not my species, and so I'm going to eat you. So they're on tiptoes trying to figure out if the female's going to be aggressive. But yeah, they'll court just about anything in the field. So, yeah, but that may play a role in some of the diversification once they secondary contact, for example, during incipient speciation and things. So, yeah.
Do we see any similar adaptations in insects regarding the red filter in the eye?
Yeah, actually, the interesting thing about this is that this is a fairly common strategy for tuning your vision. So butterflies do this. They use red filters that are along the sides of their photoreceptors, their rhabdomes, and they use that to tune green sensitive cells deeper into the red. Birds do this with things called oil droplets that are full of carotenoids that sit in the light path ahead of their retinal cells. They don't tune them quite as dramatically, but they do tune their retinas. Turtles do this. And so this is actually a fairly widespread convergent solution to how to tune your retina, your retinal sensitivities. Stomatopod shrimp, if any of you know what mantis shrimp are, incredibly colorful animals with wild visual systems. They use these kinds of filtering pigments to tune their vision as well. So it's common. This is the first time it's ever been seen in spiders. So it's neat to see this emerge again in a totally different group. Yeah. We don't have any evidence that males won't court when light levels are low. They won't court when it's completely dark. But if there's some light, males are willing to court. One of the things we're interested in is whether or not males that do better are the ones that choose to court in the right conditions. So choose to court females in bright sunlight rather than in the shade, for example. But I talked about them singing. We don't study the singing, but Damien Elias does. And one idea for why you might have so much complexity in these kinds of courtship displays is that these are redundant, so that males are using a bunch of different modalities in case one of them fails. So movement may back up color, singing may back up color and movement in situations where females can't see it, but they're on these incredibly complex vibratory environments where they're on leaf litter, and so they can't necessarily rely on females being on something that is vibratorily coupled to where they're signaling, so maybe the visual signals back up the vibration, and so there may be some built-in redundancy to these signals that allow males to court in a broader range of circumstances. So, yeah. Yeah.
This is another question on size. I work with insects under a microscope all the time, but they're easy to manipulate because they're already dead. Your student who applied the eyeliner, and I'm looking at this picture of this spider on a penny.
Yep.
How is that possible to, like, how did you apply the eyeliner and not kill it?
No coffee.
Yeah.
We have to knock these things out either with cold or CO2, so they're immobilized in that way, and you have to do it under a microscope. And I actually purchased this really neat device. It's a little micro manipulator that translates your hand movements down by a factor of 10. People use it to split up yeast tetrads. Because one of the problems that you get in doing these tiny manipulations is that there's only so much jitter you can get rid of in the human hand. But if you can translate that down by an order of magnitude, then you can do much larger movements. So this is this cool little mechanical device that just translates things down so that you can write on grains of rice and stuff with that. And
You should get that for fly time.
you should. Yeah, absolutely should. They're kind of pricey, but
If
yeah.
anybody else has questions, please come to the social. I'm sure Nate would be more than happy to address them, and let's thank him again for